So good morning, ladies and gentlemen. I would like to thank to Gessin to invite me here, and I will talk about combination of two nanomaterials in one system which is useful for non-invasive mucosal vaccination. The big opponent of vaccination was Gandhi, and it's an example, just great people is committing big mistakes. I think it's my opinion. Because still we are not free of dangerous infection diseases. We can go back to 20s, it's influenza, First War, typhus, medieval plague, and still in some countries polio, now Zika and Ebola. So there are big challenges for new vaccine development. What are disadvantages of the wholesale vaccine? It's probably safety and production safety. So there is a need for development of new vaccine, and for a long time the recombinant vaccine are target for development. And also the one problem is injection of vaccine, because a lot of people don't like injection, and there's now strong anti-vaccine movement and so on. So here is a field for development of new non-invasive vaccines. The mucosal vaccines are needed for such disease where pathogen is going inside the body through the mucosa. It's a lot of vaccines, for example, influenza. So mucosal vaccination can induce protective mucosal immunity, which is difficult to induce by injectable vaccines. The main advantage of sublingual vaccination is that you can induce general mucosal immunity together with systematic immunity. So what challenges we have to fight? It's necessary to keep antigen on the mucosal surface for a longer time. We have to ensure penetration of antigen or antigenic nanoparticles into submucosal tissue. We have select suitable adjuvants to induce mucosal immunity, and to select appropriate antigen formulation to keep native state of epitopes. So we have to develop delivery system, we have to develop adjuvants, and we have to formulate or develop new antigenic structures. The problem with sublingual immunization is that you have to keep antigen for a longer time on the surface. It's difficult because tongue is moving, the salivary glands are producing the fluid. So most of antigen is removed from mucosal surface, and mucosal system is not in interaction with the antigen. It's normal, it's natural. If antigen is for a longer time on the surface, it means for immune system that it's danger, because there's a tendency to penetrate into the body. And that's why we need some system which can keep the antigen for a longer time on the surface and to mimic some adhesion. Sorry, I haven't a pointer, so I cannot describe the situation. But you can see on the top antigen forming some gradient, and the gradient can increase penetration of the antigen into submucosal tissue. We have developed this mucoadhesive films, and the heart of the film is a special material. It's a non-woven nanotextile. It was developed, the material was developed at Technical University in Liberec, and we are collaborating with this group. And we have applied 2 patents for this system. The system ensures 3 things. Long-term deposition of antigen on the mucosal surface. It prevents penetration of the antigen outside the mucosa, and it's a reservoir for antigen, adjuvants, and maybe penetration enhancers in one very thin layer. Here is some example of application of these mucosal films onto sublingual mucosa or buccal mucosa. Sorry. It's important to say that in sublingual and buccal mucosa, there is a lot of dendritic cells, and these dendritic cells have different function. One are responsible for inducing energy, so you are not responding to a lot of antigens from the food, and other are responsible for inducing immunity if the antigen is applied with some molecules which are called danger signal molecules. So now I will talk about nanoliposomes as a carrier for construction of recombinant vaccine. We can skip this general information. Yes, liposomes are only one nanoparticles approved by FDA and EMA. No other nanoparticles are approved at present. So we are focused on these particles because they are biodegradable, they are approved. There's a lot of technologies how to produce these particles, and these particles are produced by pharmaceutical industry. And another advantage is that liposomes are very versatile. So we can combine liposomes with different type of adjuvants, molecular adjuvants, and we can apply liposomes both by injection or topically. It means also on the top of mucous tissue. We have pioneered the metallochelatic liposomes as a platform for self-assembly system for recombinant vaccine. So it's a very simple system. You can prepare liposomes with metallochelating lipids. A lot of antigens or recombinant proteins are produced with his-tag. So his-tag can interact with metallochelating lipids, and it's non-covalent but relatively firm bound. And you can add also different type of molecular adjuvants which are necessary to enhance the antigenicity of your antigen. So this is very simple system which can be produced in a large scale. In the past, we designed and constructed several devices how to produce defined liposomes. So it's one device for detergent removal method, another is for detergent removal method by ultrafiltration. Especially this system is linked with old FIFO FPLC system, and we can develop automatization, high degree of automatization. You can add antigen after forming liposomes and so on. Another system is for liposome method. We are using different secondary processing methods like high pressure homogenization or high pressure microfluidization. Also we are using extrusion and high pressure extrusion for 50 milliliters. Here I would like to point on one thing. If you prepare liposomes by extrusion, there is a very high portion of liposomes which are not unilamellar but multilamellar or oligolamellar. So, but you can tune the size precisely from say 200, 300 nanometers to say 50 nanometers. We introduced this year also the nanofluidization mixing to produce liposomes, and we tested to produce just empty liposomes suitable for surface modification. It's my student. And we developed the platform which is based on different lipid composition. This is one example how we can tune the size distribution. And it's very important that we can reach very low polydispersity from 0.05 to 0.02, and we can tune the size. This is one example of liposomes with adjuvants. This is trehaloids dibehynate. At present, we have synthesized in collaboration with the company Apigenex about 60 different derivatives of this compound to find the most suitable adjuvants. And it's important to see, I think, in comparison to, here we can compare liposomes prepared by extrusion and by nanofluidization. We can see the same distribution, very narrow distribution, but you can see that on the left, the intensity is very wide. So it means that it reflects internal multilamellarity. And extrusion is producing multilamellar or oligolamellar liposomes, but nanofluidization produces strictly unilamellar liposomes. So you can, it's reflected also by the distribution of the intensity, not just by size. So we have all technologies from lipid to lyophilization to produce various liposomes in a laboratory scale. And now I will talk a little bit about adjuvants, which are very, it's a gold grail of vaccinology now. For a long time, we are working in collaboration with Dr. Ledvina and Andy Miller on monomer glycopeptides derived from peptidoglycan. At present, maybe it's history, more than 50 years history, only 3 compounds are close or reached the market. I think monomer tripeptide is only one used in therapy of osteosarcoma, but not as adjuvants, because as adjuvants it's not safe. We have developed different type of muramyl dipeptide-based analogues, which are strictly apirogenic, and we at present they are protected by US and European patent, and we have a set of different molecules. These molecules are suitable for incorporation into liposomes because of hydrophobic function linked with sugar or peptide. We also proved the molecular mechanism. These compounds are able to activate NOT2 receptor, which is a receptor for monomer dipeptide also. So on the molecular level, probably they are using the same signaling pathways like the parental compound. And in liposomes, they are they are better for delivery to immune cells and to induce NOT2, and recently we have found also that they are able to induce NALP3, its inflammasome. So if you add the antigen in this appropriate adjuvants in the same particle, you can induce efficiently both Th1 or Th2 immune response. So at present we are working with different adjuvants like monophospholipid A, trehalose dibehenate, NOR-ABU and NOR-ABU GMDP. And also we can incorporate into liposomes some hydrophilic molecules, which are generally TLR7 agonists, and they are produced by different companies now. So now I will talk about this concept and the joining the concept with nanofiber material. Here is some example of cell-assembled vaccination nanoparticles. I think the interesting picture is the red one. It's from atomic force microscopy. On the left side you can see Bohr liposomes. On the right side, B4, you can see nanoliposomes with bound recombinant protein. In this case, it is heat shock protein derived from Candida albicans. And what's important, these particles are able to induce the immune response comparable with foreign adjuvants. So you can see on the left side it's boosted. The yellow column is for liposomes adjuvanted with one derivative of MDP, and the blue, violet, and orange is various combination of foreign adjuvants. So we have system which is very immunogenic and can increase immunogenicity of purely immunogenic antigen. it's important to say that this system is able to strengthen Th1 immune response in comparison to alum. You can see that alum is able to induce IgG1, which is typical Th2 immunoglobulin, but our liposome with MDP analogues can induce especially EGJ2A and EGJ2B, which are typical Th1 in mice, Th1 immunoglobulins. And at present, this system was used and is under development in the company Bioveta, it's a Czech company, to produce veterinary vaccine against lymph disease. And now we are working on the vaccine for human. For veterinary vaccine, probably alum will be used because it's cheaper and they are familiar with these adjuvants, but for human application, we prefer liposomal formulation. It's an example of the vaccinated nanoparticles. The black dots are nanogold with antibody against OSPC, which is bound on the surface of this liposome. So we are going back to mucosa application. So we can formulate liposomes with antigen. We can formulate it with adjuvants, and now we have to apply this liposomal antigen onto the surface of sublingual mucosa, and we can use this nanofibre-based mucoadhesive films. This is a nice picture from confocal microscopy and electron microscopy demonstrating liposomes, the small spherical objects on the surface of nanofibers. So we are able to adhere liposomes. We can modify the surface of nanofibers to tune well the adhesivity to different particles, nanoparticles, and also we can modify the surface of liposomes to reach the same task. The system is very robust, and we can incorporate not only nanoparticles but also, for example, bacterial ghosts, which represent another very modern system for vaccine development. So you can see the green objects in the red mesh are the bacterial ghost, and you can see then human dendritic cells, which is able to entrap the bacterial ghost. Another example is showing nanofibers with plasmid. So this is a way to DNA vaccination, or we can formulate plasmid also in liposomes, and it's more advanced system for DNA vaccination, or we can combine DNA particles with antigen, so it's in one particle you can reach both DNA and recombinant protein vaccination. It's a modern approach. Another example is showing virus-like particles derived from bovine papillomavirus and characterization. And here it's not well resulted, but here is a herpes virus adhered on the surface of nanofibers. And here again you can see internalization of fluorescence nanoparticles adsorbed on nanofibers to human dendritic cells. The red is the fluorescent nanoparticles. And biocompatibility is also very high. Here you can see how dendritic cell is going through the mesh of this material. Another application of nanoassembler is for preparation of. Finished? Yeah. So I will go quickly. So nanoparticles based on PLG. We are close to the finish, and we can skip it. PLG nanoparticles, and what is important, we used PIC models to show that nanoparticles really penetrate into the mucosa. So on the left you can see penetration of fluorescent nanoparticles. Here is tight connection of nanofibers with the submucosal tissue, and this is important. Here you can see massive penetration of nanoparticles, and paracellular penetration is clear here. And what is the most important part, we can find after 1-2 hours nanoparticles in draining lymph nodes. So it's a prerequisite to induce immune response in both T and B areas. And conclusion, this is a pathway to technology we now calling printed vaccine. And the technology is developed in collaboration with Daniela Lubasová. This is an instrument for preparing industrial textile, and here is the first attempt to print antigen on the nanofibers. So you can see that we can print precisely different type of liposomes with different antigens. So multi-vaccine development is feasible. And now we are focused on influenza because it's, I think, the best and the right target for our technology. So we can skip this one. My collaborators, my team, Dr. Ledvina from the Institute of Organic Chemistry and Biochemistry, responsible for synthetic work, Professor Kratochvílová and Atomic Force Microscopy Group, Nigel Temperton from University of Kent, Professor Sherman from René Descartes Institute, and Professor Miller as a long-term collaborator. And that's all. I think I would like to invite you next year to Telč. It's a city under protection of UNESCO, and we are organizing a very small meeting for 80-100 people. This year it's 10th meeting in a row. It's focused on vaccine and new technology. So you can enjoy also personally as a musician. So thank you.
In view of the time, I think we should press on very quickly. But Jaroslav, we're sure there's at least 1 or 2 questions we can take right now from anybody who would like to offer a question to that presentation. Okay. Jaroslav, would you kind of come to
Yeah, just a quick one here. For the last few slides on the penetration in the tissue, did you see any impact on particle size and penetration?
The impact on particle size on the penetration. In the last stage of in terms of the mucosal penetration, I think there's still work to be done on that. These were standard size PLGA-particles. So that is something that, yes, we would do. And of course, the nanoassembly would be perfect to be able to actually get a size range check and do those kind of studies. Yeah,
okay,
absolutely.
Thanks for that.
Okay. Anybody else?
Why choosing, thank you. Is there any reason why choosing pig models for mucosa vaccines?
It's a good question because only pig models is reflecting anatomical similarity with human. Because the same food is prerequisite for the similar anatomy. In maybe mice, it's 300 microns thick, the mucosa. In human and pig, it's 50 microns. So it's one thing. Second thing, it's not keratinized. In rodents, the mucosa is more keratinized than in human or pig. So it's a very good model. You can quickly obtain lymph nodes because in mice it's 0. And you can also study transfer of maternal immunity to offsprings. So it's a better model and more resembling situation human.
Thank you very much. Could I just say, just before I introduce the next speaker, that clearly what Jaroslav has presented is a platform. Yes, of course, there's a lot of vaccination nanoparticle technology that we've also been able to build up over the years, and Jaroslav is very expert in that. But this is actually an open invitation as well to talk. those of you who have vaccination nanoparticle systems and you would like to investigate alternative means of delivery in a non-invasive manner, then I'd absolutely be interested to talk. The platform is very flexible, will contain a wide number of shapes and charges, so therefore, shall we say, if anybody's interested to follow up with us afterwards, either myself or Jaroslav, that would be much appreciated and would be very interesting. Okay, thank you very much for that.